Today's objectives are to continue to identify areas of probability in genetics and learn how to solve a dihybrid test cross. In our previous lesson, we took a look at Punnett squares, or monohybrid test crosses, where we just tried to solve for one gene. Today we're going to look at Punnett squares in the form of dihybrid test crosses, and this is when we look at two genes. For example, we're looking at round yellow seeds versus green and round seeds, wrinkled yellow seeds and wrinkled green seeds. So we have two different things going on here. We have round versus wrinkled and yellow versus green. In this lesson, I will show you how to fill out a dihybrid test cross. It's a little bit different than in a Punnett square, and usually with some practice you'll get the hang of it. So let's begin to set up a dihybrid cross, which is something that shows the possible offspring for two traits. When we're looking at color, which is yellow, capital Y, and lowercase y is green, and shape. Uppercase R is round, lowercase r is wrinkled. In this case, we will cross a heterozygous individual with a homozygous individual. Their genotypes will be big Y, big R, little r, and the other one will be big Y, little r, little r. When we set up a dihybrid cross, we have to look at both parents and we have to list the possible gametes. And there's a particular order to this, but we'll go over that very shortly. A gamete is a sex cell. In a male, it's sperm. In a female, it's an egg. And in each one of these sex cells, we need a Y and an R in there. There needs to be one Y and one R in there. And there's the combinations are the ones that are possible in each of the parents. So our several possible gametes are big Y, big R, big Y, little r, little y, big R, little y, little r. What we're going to do is we're going to arrange these gametes for one parent along the top of the Punnett square, or the test cross, and all the possible gametes for the other parent down one side of the Punnett square, or test cross. We're going to use a process called FOIL, which stands for first, outer, inner, last. We're going to take a look at our first test cross here, and we're going to set it up using FOIL. We're going to take a look at big Y, little y, big R, little r first, and we're going to use FOIL, which is first, outer, inner, and last. The first parent here we will place up top, the second parent we'll place down the side. And remember, we're looking at the different allelic gamete combinations that we could have here. So our first stands for the first Y and the first R, and we'll put it up top. Outer is the outside Y, the outside R. Inner, the inside Y, the inside R. And last, the last Y and the last R. And we put those all up here. Now when we do the homozygous parent here for both the green versus yellow seeds and the round versus wrinkled, we will get the same allelic combination no matter what we do, and that's big Y, little r, because these are the only two combinations that are possible in this parent. Now that we have the dihybrid test cross completely filled out, we can start collecting data. And the data will be collected by answering the following 4 questions. And the questions are based on the phenotypic outcomes that could happen. How many of the following offspring would have yellow seeds and round peas? That means it has at least one capital Y and one capital R. There are 8 of the outcomes that do follow this big Y and a big R. Therefore, 8 out of 16 will show yellow seeds and round peas. How many of the offspring would have yellow seeds and wrinkled peas? So yellow is capital Y, and wrinkled, we must have 2 little r's. If we follow Mendel's principle of dominance, it says that the recessive only shows when the dominant is not present. So we have 8 scenarios where there's a capital Y and 2 little r's. So 8 out of 16 will show yellow seeds and wrinkled peas, which means that there are 0 offspring that will have green seeds and round peas, because we've used all 16 boxes and we classified them, and there will be 0 out of 16 that has green seeds and wrinkled peas. So to double check yourself, you should add your 2 numerators here, which equals 16, and make sure that it also equals the denominator, which is 16, which it does. So this is how we would fill out a dihybrid test cross, and we would gather our data from it. So what we do is, from here, is we come up with a phenotypic ratio for all 4 outcomes. In this case, it's 8 to 8 to 0 to 0. It just means that there are 8 that will have yellow seeds and round peas, 8 that will have yellow seeds and wrinkled peas, and 0 that will have green seeds, round peas, 0 that will have green seeds and wrinkled peas. So if we go over another dihybrid cross, we have 2 traits: fur color, which is black and white, and coat texture, which is rough and smooth. In this example, we will cross 2 heterozygous individuals. Their genotypes will be big B, big R, little r. Alright, so this is our dihybrid test cross for the heterozygous parents. There are several possible gamete combinations: big B, big R, big B, little r, little b, big R, little b, little r. We're going to use the same foil to set up the test cross. What I'd like you to do right now is to pause the tutorial and set up your test cross using these 2 parents. The first parent will be along the top of the test cross. The second parent will be down the left side of the test cross, just like we did on our previous dihybrid test cross. When you are done setting up the gamete combinations, you can resume the video, and just to double check to make sure you had it right. Alright, so I use foil and I set up both of the combinations for both parents along the top and along the side. At this time, just double check to make sure that you have the combinations right, and then complete the dihybrid cross in here, and then continue on with the tutorial. Hopefully you were able to come up with the correct combinations for the test cross. Please make sure that you have those correct. The next thing that you'll do is you'll go through each one of the phenotypes and write down how many of the individual outcomes over here will be represented by each one of these phenotypic outcomes. When you're done with that, continue on with the tutorial. If you have any trouble with that, try to come back and see what you might have messed up on inside of the test cross. Alright, so here are the answers to each one of the questions. I did them in color code. For the first one, black with a rough coat, there'll be 9 of them, as you can count. 1, 2, 3, 4, 5, 6, 7, 8, 9. Black in a smooth coat, 1, 2, 3. White with a rough coat, 1, 2, 3, and 1 white with a smooth coat. So all in all, our ratio should be 9 to 3 to 3 to 1. So in today's objectives, we did identify an area of probability in genetics. Last tutorial was a monohybrid test cross, or a Punnett square. This time our Punnett square was a dihybrid test cross. We did learn how to solve a dihybrid cross. We will do more practice in class. If you have any questions or any areas of confusion, please make sure that you bring those up at the beginning of next class.